Okay, guys, this is a lecture on Chapter 8, and that means that the lecture on Chapter 7 and the lecture on Chapter 8 will represent really the first part of the Exam 3 material. So you guys should interpret this just like you are sitting right there taking your own notes. The cool thing is that you can go back and forward if you need to write something down. I'll probably be going a little bit faster than usual because I don't have an audience to kind of get my attention. So it's a pretty intense chapter. It's really important to me that you do both the Chapter 7 lecture on video and the Chapter 8 lecture on video and make your notes for that before we meet on the Thursday after spring break. So we won't be in class on the Tuesday after spring break. That's what this is for. And then on the Thursday after spring break, we'll briefly evaluate what I did on this lecture, and then we'll just keep moving forward, and we'll be on Chapter 9 on that Thursday. So this is a chapter about energy, and I want to kind of set us up for this abstract concept of energy. We're going to examine energy in chemistry primarily as heat, and the fundamental unit would be calories. And that's pretty much ingrained into all of our brains. We think about certain kinds of exercise that we do and how many calories that we burn. I want to think in energy in terms of a more visual way. Repulsion of like charges is energy, isn't it? So I'm pushing these guys together and it's kicking back. That's kind of a visualization of energy. One way to look at energy in your book is to break it down into two different kind of categories of energy. We have kinetic energy and we have potential energy. So the simplest definition of these would be that this is energy of motion. And this is energy, or this is stored energy. So energy of motion and stored energy. What your book likes to do is to present this classic, so here's a hill, and up on the hill is a big stone. This is a very round stone, and that particular stone in its position is tons of potential energy, PE. So let's do PE, KE, and then somebody pushes this guy, pushes it down the hill, and this part here is the energy of motion. So this is the KE. So the potential energy has to be released. A lot of times potential energy has to be realized as kinetic energy, energy of motion, energy to do work is what they say, requires that some amount of energy is put into the system. We're going to look at this in a better way as we move forward. Hope you can make that out on the board, but that is my little cartoon of a strong person pushing this boulder down the hill. And then, of course, once it gets there, it goes. So kinetic energy and potential energy, we want to keep those in our mind. Then we want to break energy down in terms of the kind of the modalities, the modes of energy. And I'm not going to give the whole categorical listing one by one, but we have mechanical energy. We have heat energy, we have sound energy, we have electric current, and then we have radioactive decay, radioactive particle, so I guess I just call it radiation. These are all modalities of energy that are present. And I think that the one that I want to put a star by in biology is that our standard measurement in biology of energy, in bio or biochemistry, we measure energy. The unit of measure is something called the calorie. So it's heat energy, and the unit is the calorie. The calorie is going to be defined down here as the heat required to raise 1 gram of H2O 1 degree Celsius. So this is the amount of heat required to increase 1 gram of H2O by 1 degree Celsius. That's what a calorie is. Now, we've already talked about this kind of thing before with regards to specific heat of water and how it was more resistant. It didn't increase in temperature, so different kinds of different amounts. But the heat is called the calorie, and of course, we want to always realize as well that 1,000 calories equals 1 kilocalorie. Interestingly, the calorie that we talk about on our nutritional labels is actually, we say calories, but it equals kilocalories. So if you say, I'm going to take down a Snickers and it's got, 352 calories in it, it really means it has 352 kilocalories. So a lot of energy in a Snickers bar. So heat is the measure in biochemistry. Let's take a look at the real big picture. I want everybody right now to just completely focus on this. This is the big picture, and it will define what we do in this class for several chapters. It'll set many lectures. We're going to start with this energy one, and pretty soon we're going to be right in the middle of the mitochondrion, figuring out part of this. And then after that, we'll be right in the middle of the chloroplast, figuring out the other part of it. It is the big picture. I call it the big picture. What can I say? So the big picture is this. We're going to take our friend CO2, which of course is a gas. You know what this is. You can draw the structural formula of the things, I'm sure. And we're going to take H2O. You can draw the structural formula of that as well. And we are going to convert that into this. And you can draw the structure of that and that. You guys can draw the structure of all 4 of those molecules. What we're doing, however, the most important thing is that we are making covalent bonds between carbon atoms. Those are going to be our future gold mines of energy. We're going to break those bonds, starting with glucose in the mitochondrion, to ultimately produce ATP. That's pretty cool. Carbon-carbon bonds in that molecule are what it's all about. So, I'm going to write down in red. This molecule here, which of course we identify as glucose, or certainly a monosaccharide, is full of, or it has, many high energy carbon-carbon bonds. And of course, those are single covalent bonds in that molecule. So overall, to do this process, I'm going to get green out and identify this as photosynthesis. And I'm going to highlight this arrow as being the arrow of photosynthesis. And that process is going to require energy from somewhere else. So here's the somewhere else. It's the happy sun. And the happy sun is actually flooding down on the chlorophyll pigments inside of the chloroplast. That's coming up. And that energy is being transferred This is going to therefore be what we could call it solar energy, call it light energy. And what we really want to say is that if we think about this in terms of particles, photons, that's what it is, photons. These are these theoretical particles of light, energy. So the sun is showering and putting this form of energy, this light energy, and converting it into this energy. We call this chemical energy. So this thing, let me get another color to show you that. This energy, well, let's see, that energy right there is what we call chemical energy. Whoops. So here we have solar energy, solar or light energy being converted into chemical energy in this process that I've identified as photosynthesis. The other thing I need to tell you is that this is a grand overview of many reactions. So each one of this arrow requires many, many steps, many individual chemical reactions that lead to this overall process. The other way is what we'll talk about actually first. This guy I'm going to highlight in red, using red and green to codify mitochondria and chloroplasts respectively. So this is what we will call respiration. So I guess you should remind ourselves, chloroplast. We should remind ourselves here that this thing here happens in mitochondria. Underline those because those are important. And that is the process. Okay. That is the big picture, you guys. So this energy is going this way. This energy, so we have energy being increased in the forms of covalent bonds. This thing is providing energy for work. And in general, this will be in ATP. Which we will define carefully later in this lecture. ATP. I want to get used to doing one thing when we do ATP. ATP is energy. Orange is going to be my energy color. And I'm going to put a graphic novelist pow. You know, like when somebody hits somebody in a graphic novel, there's like a big starburst of energy. Well, there you go. ATP is going to get a pow for us because it represents biochemical energy that is ready for action. More on that in a minute. So this is the big picture. This is a super important part of this entire chunk of the class. This is the entire unit, really. So this process going this way, this process going this way, two different things. This one has energy in, this one has energy out. At this point, the energy in ATP is still chemical energy, but it's a different kind. So this energy here is chemical energy. So here we have these different kinds of energy. That's going to bring us to a funky field of thermodynamics. Light energy, chemical energy, chemical energy. So something is going on here. This is what we call a energy transformation. we're going from one type of energy, light energy, to another type of energy, chemical energy. And this is also an energy transformation. Because here we're going from one form of chemical energy, carbon-carbon bonds in a monosaccharide, to another form of chemical energy, which would be what's called the phosphodiester bonds of ATP. Let me write that down. So this Okay, so I've written this down. So the chemical energy here is in the energy of ATP is formed in the form of the phosphodiester bonds. This is the name of the covalent bonds that forms between the phosphate groups. You should probably also draw a pretty good phosphate group for us. So that's the big picture. Take a look at that. That's amazing. What we need to do now is carefully consider energy transformation. The world of energy transformation, this whole concept, is part of the field of science known as thermodynamics. We need to understand just a couple of laws of thermodynamics. And I'm going to erase and get ready for that. Okay. So, we need to concern ourselves with the rules of energy. And there's two that we need to look at. The first one is the first law of thermodynamics. Of course, I've already said that in biochemistry or in science in general, that the units of energy, the calorie is a measure of heat. That's where the thermo part comes, and the dynamics of heat. It's the dynamics of energy. The first law of thermodynamics, which you must know, I will double underline it, is that energy cannot be created or destroyed. Cannot be created or destroyed. Energy can only change from one form to another. So what I want to say is changing form, aka transformation. Change from one form to another. Extend that underline. So that is it. There's another part to add to this. The total amount of energy in the universe remains constant. Okay, that's the first law of thermodynamics. Remember it, it's super, super important. You can't create energy. Energy can only be transformed. And the general flow, of course, of energy is going to be the next one. The second law Second law of thermodynamics is that entropy, which will be a term for us, entropy, which is disorder in the universe, is continuously increasing. There we go. That's the second law of thermodynamics. What you'll see in your book is this interesting idea. Think about the room in your house or your bedroom, whatever, that you've cleaned, and watch it become progressively disordered. That is the second law of thermodynamics in an analogy. The same idea would hold true for a substance. This is pretty cool stuff. Let's think about a drug. So that's a very, very focused kind of example of entropy. You take a drug in its form and just put it on the shelf in the bottle. It has a date on it, and that date is kind of a use before date. Maybe it's an antibiotic, maybe it's some other targeted drug. And that drug is going to change without anything else, without any other influences. Just sitting in a bottle on a shelf, it's going to change through the course of time to the point that it is perhaps not even functional anymore, or marginally functional compared to when you just got it from the pharmacy. That's an example of of disorder, molecular disorder, from something that was very ordered at the beginning, continuously increasing. So we're going to wrap our brains around these as we go further into the concept of energy. I'm telling you though, right now is the big story. It's the energy graph and identifying two kinds of reactions. One, where reactants are higher in energy than products, the other vice versa. It's big stuff, so I hope you're ready to march ahead. So we're going to take a look at a typical energy graph. The first thing that we want to talk about, the first variable, is going to be on the y-axis. We have G, and G stands for free energy. Therefore, delta G is going to be something that is going to be the change in free energy over the course of a reaction. There's my abbreviation. Okay, so that's delta G. And the other thing that we need is T, and T is going to be time. So on the y-axis, we'll always place G, and of course, it's high at the top of the axis, low at the bottom. And then we'll have time. Time is increasing to the right. That's the basic graph, x, y, two-coordinate system that we use. So to describe a reaction. Here is a reaction where we start at some point in time with the reactants. Let's say that this is 0, so we'll say that right here we have the energy of the reactants. And we'll watch this graph occur like so. I'm drawing it as it would be, and then it reaches a steady state down here. This is the energy of the products. So a couple of things to say about this. Let's draw a nice dotted line there. This is the energy from that dotted line to there. That in this particular case, this is high, this is low. This is energy given off by this reaction. We would say here that the delta G is positive. Therefore, we should figure out how to calculate delta G. I'll do this up here. So G of the products minus G of the reactants equals delta G. So that's G of the products, excuse me, the reactants. G of the reactants, good grief. Sorry about that. G of the reactants minus G of the products. And is equal to delta G. And if that is a positive value, because this number is bigger than this number, then in this particular case, we've got something to say about this reaction. So I'm going to change that and introduce this. When this occurs, when delta G is positive, this is called an exothermic, aka exergonic reaction. Energy is given off to the system. This energy that is given off could be for work. Work is KE, kinetic energy, etc. So this is a reaction where something happened chemically, the reactants going to the products, and energy is given off. There's an energy transformation in here as well, then, of course, because we've probably broken covalent bonds and released covalent bond energy, which is called chemical energy. And if we're doing work, we're actually doing mechanical energy. That's an energy transformation between the modes. That is what we call an exergonic reaction. There's a ton of hard information here on the board. The first thing is to really get this thing down. X and Y, G, time, and then identifying reactants and products in the curve. We've got something else to say about this, and I'm going to say it in a different color. This is a very important little piece here. You can see that before this reaction occurred, as time flowed, energy was put into the system. This is what we call E sub A, which is equal to activation energy. And it is the energy that has to be put into the system in order for it to occur. There you go. So that is a full, brilliant, and beautiful exothermic reaction. If I'm not mistaken, back here, your textbook's going to use exergonic, and your lab manual, perhaps, is using exothermic. I'm a big fan of exothermic, and take a chemistry course and you'll see why. This is what is held on to, by all accounts, for your stereotypical chemistry courses. Good stuff. Energy graphs. Now, I've got this one standing. I'm going to squeeze another one in that shows the other possibility. Here is a reaction down here. I'm going to try to cut this off. So the rest of the board, I'm going to show you another type of reaction. The other major type. Here's time going on. Here's G. And we're going to start low with our reactant energy. And we're going to go up here like so, and we're going to end here with our stable energy. I'm just going to go here. That's what we're going to do. So this is our energy, this is our products, reactants. So now we've got this whole chunk of energy that you put in. Put into the system. this is very interesting. This also equals E sub A here, and this reaction is defined with products having a higher energy than the reactants. Check this out. G products minus G reactants is equal to, in this case, delta G negative here. And we will call this reaction with another color. This thing, when this occurs, it is called an endothermic reaction. And of course, that is AKA endergonic in your text. Endergonic reaction. Two different kinds of reactions. Okay, so I hope that this makes a bit of sense. Reactions are going to be either exothermic or endothermic. It's simply the way that it is. The next step is for us to see these reactions being coupled. So an exothermic reaction can release energy that can be coupled to drive an endothermic reaction. And that is the key to understanding biochemical reactions. So let's take a look at the big picture. All right, here we go. I've been promising that we're done with structural formulae. This is going to be part structural formula, part cartoon, but we need to do something. We need to celebrate the extraordinary structure and function of ATP. So ATP, of course, is a nucleotide basically, and let's stick this together. Ask you guys some questions to refresh your memories. So first off, sugar, correct. Here's a shug, and here's a base. Purine or pyrimidine? It's another question for you to solve. Purine or pyrimidine, because it's the same language that we dealt with before, same concept. We're going to stick to this off of, again, let's number our carbons just for fun. Just for fun, memory lane. Number our carbons. Carbon 1, whoops, let's get a better pen to number our carbons. Throw that one away. Maybe. 1, 2, 3, whoops, 4, 5, could that be it? Wow, that's pretty cool. So, and then of course you guys might be thinking DNA. 5 prime, 3 prime, 5 prime, 3 prime, 5 prime, 3 prime. Sorry, we'll come back to that later. So, here's our base, here's our sugar, and now comes the fun part. Let's draw our phosphates, structurally, however. Oxygen, phosphorus, oxygen, minus charge, double bond oxygen, boom. And then, keep your color handy. I am going to draw one of my covalent bonds here, red. And then I'm going to set up yet another of our phosphate groups with a full negative charge there, double bond oxygen here, oxygen, and then another red guy, and then phosphorus, and then let's hit these, oxygen minus, double bond oxygen. Boom! That thing rocks. It rocks so much that I'm probably going to have you memorize this for the test just for fun. We have identified with red now some very important things. First off, this whole linkage here is a phosphodiester linkage. I'm going to change from blue for that label. The key for us is these bonds right here are high energy covalent bonds. Understand that and know that. Those are extremely important. I want to take us to a new concept here. Not only are they high energy covalent bonds, but these guys they're short fuses. These bonds are more likely to break than a lot of bonds, and I'll tell you why. I hope that you can get this. So, negative charge, negative charge, negative charge. If you think about that, like charges repel. Opposites attract, so positives attracted to negative. Like charges, such as negative to negative in this case, repel. So these guys are pushing away from each other. There's a lot of hate here. This guy wants to go this way, this wants to go this way, this wants to go this way, this wants to go this way. This whole thing renders these bonds stressed. Are stressed by the repelling of the negative charges. Because of this, these are pretty easily given off. They're pretty easily broken. Relatively easy to break. And when you break a covalent bond, a chemical bond, that energy's got to go somewhere. Energy, thermodynamics. This form of energy, you break a bond, it's got to go somewhere. And what kind of bond do you think, or what kind of reaction do you think it is when we break one of these? It's a give energy off kind of reaction, aka exothermic reaction. So this guy can break down and give off a phosphate group and give energy to a system. It will be transformed into doing work or becoming another bond, or as part of a reaction. It's that kind of stuff. So take a look at this. If we take ATP and we do this reaction, here's the reaction. So this is the reactant. Here's the product. Whoops. No change to this business. Same old story. I'll leave that one intact. And boom, plus now we've lost one of these guys, which a lot of you in your exam drew that phosphate group like that. So we've broken a bond. This is ATP, which is the R of the reaction. These are the P's of the reaction. And this guy here, this big guy here, we call ADP. This guy we call P sub I. So ADP plus P sub I are the products of the breakdown of ATP. Here's the graph, GT, and here is ATP, ATP, which of course is a reactant. This reaction has gone this way and produced our P's, which would be ADP plus pi. In the process, we have given off this energy given off. This, of course, to remind you, should be clear to you that this is an exothermic reaction. My goodness, look at that. So this is what we're going to do. We are going to have ATPs in the system. ATPs are going to be broken down into ADPs, and that is going to make, and plus inorganic phosphates. And that is going to drive other processes because this is an exothermic process. The reason that it's exothermic is that one of the reasons it's pretty easy to happen. These are stressed bonds. Stressed bonds. Breaking bonds is usually exothermic. Making bonds is usually endothermic, okay? And these guys are stressed bonds that are easy to break. This is our energy source for everything that we do. Those Dynean bridges that we talked about when flagella were doing their thing, remember that? Changing form. Those Dynean bridges are changing form because of ATP. More on that later. Excellent. This story's not over. I can go from this process here to, here's my energy graph, G and T. I'm going to get the same kind of energy here. Here we have the product being ADP, or excuse me, the reactant. Here we have the product being, guess what? AMP plus pi. Circle those in blue so they stand out. So we can draw this guy down here. My drawing's going to have to be smaller because of the size, the space that I have left, but let's do it anyway. There's our sugar. Okay, so then we start with phosphates, P, O minus, double bond O. This, my friends, here is a MP, standing for monophosphate, plus another phosphate group that got busted off. Plus PI. So there we have it. I'm going to draw kind of a line to show how this thing took place. ATP breaks down, exothermic reaction gives off, it becomes ADP plus pi. ADP then breaks down to become AMP plus pi. So this is D for diphosphate, M for monophosphate, and then finally T for triphosphate. Check it out. The big energy driving force in biochemistry is on the board now with energy graphs, words to go with. Let's write down the word exothermic. To remind you of that, of course, this is the energy given off. Exothermic. When the reactants have higher energy than the products, energy is given off to the system, we call that an exothermic reaction. Wow. So there's ATP. Now, we're going one step further. I'm going to show the coupling of reactions. Here's how it works. Okay. So now let's take a peek at this in a simplistic way. ATP, which I should have done this on the previous board, POW. I'm going to give it the POW symboling now. And I'm going to show you that ATP lower. This whole board I want to set up on the energy scale. So this is basically the big picture of G, of energy. And we're going to describe it as high here and low here. So this is our spectrum. ATP is the highest energy on this particular family, and it's going to break down to ADP plus PI. And the reaction that's associated with that, I'm going to draw right here, just like this, just a kind of a little, you know, GT snapshot. Here's the reaction. And the same thing would hold true as previously discussed to describe the process of ADP breaking down to become AMP. That is going to be the same kind of exothermic reaction. So let's draw that right here. So there's a lot of stuff right here. Let's discuss now how to make ADP from AMP. This is going to require energy to be put in, so I'm going to use blue to show that this is an endothermic reaction, G versus T, to make ADP or to ATP from ADP, same thing, G, T, put energy in. So that's how it goes. Energy graphs show the process. Energy, or excuse me, thermodynamics should be just boom in your brain all the time. This is chemical energy that could be converted into something else. Let's show the coupling now. So the big title of this is going to be the coupling. Energetic coupling of reactions. And man, it's actually let's call these biochemical reactions. Biochemistry is the chemistry of life. So that's the big picture of ATP. Isn't this an awesome system of energy using a recycled substrate? The energy carrier here, ATP, ADP, AMP, is this basic substrate. It's basically add a phosphate, take a phosphate for the whole dynamics of this energetic system. Very, very conservative and recycling oriented. So let's take this energy here. We need some sort of a color that flashes to us energy transformation. Don't know which one to use. I guess orange is probably still the best. So here's our energy transformation. This way, energy transformation given off. That's going to drive another reaction. This energy could be put in to drive, to drive an endothermic reaction. So, G and T. And we're putting this energy in here. Let's go here. Also, as well, driving this reaction. So this sequence of breakdowns of ATP to ADP and AMP respectively is providing energy to drive endothermic reactions. So remember this. And guess what these reactions are? This could be a biosynthesis step. You know, we need energy to do these biological reactions. We have to put energy in this. Things just don't happen spontaneously. We have to put energy in. This could be basically the shape change of dynein. Example, shape change in dynein. that's a reaction. Other things, example of these endothermic reactions, a biosynthesis reaction, where we're actually forming covalent bonds, but we have to bring things together. ATP is part of this. So that's excellent. We need to look at it from this perspective too. We need to take reactions that are exothermic, like this one here, and we need to give this energy given off to drive the synthesis of ATP. And there would be another reaction here that can make ATP. So the orange here is the coupling. The orange here, these orange squiggly arrows indicate the coupling of reactions. Here are exothermic reactions. An exothermic reaction, something where energy is given off to the surround, and that energy is used to drive the synthesis of ADP from AMP. I'm going to get into the ways that ATP is made in the next chapter, the specific ways in which it's made. It's really cool. There's a couple of different ways. We're going to see that happen. But right now, I just want everybody to know that this is kind of the energy intermediate kind of transforming step. ATP, ADP, AMP is the key to the understanding of the big picture. Everything that happens in biochemistry is making and breaking and making and breaking and making and breaking. We do a lot of making. We do a lot of breaking. You think about what's going on right now with the glucose that was in whatever you fed on, whatever you ate sometime during the day, and we're breaking things down. That glucose, if you go back to the original big picture reaction that I showed at the beginning of this lecture, that thing is going from, if you follow the red arrow, the metabolism arrow, we went from glucose or some monosaccharide to CO2 plus water. Breathe it out right now. We broke down a whole bunch of stuff to finally get that CO2 that came out. This was involved in the middle of that, I guarantee it. Coupling of reactions. Give off to drive. Give off to drive. Okay, all right. So now we're ready to work into the other cool part of energy, and that is special molecules that can aid in reactions occurring. So let's look at a typical energy graph. Here's G, here's T. We've drawn this one before, and we said, okay, that's the energy of the reactants right here. The energy of the reactants, that's the energy of the products. And then I specifically drew this bracket from this parallel line to here to show that given off. This is the thing that we defined. I'll get my green pen again. We defined this as the EA, and it's called the activation energy, and that is defined as energy that must be put in. Put into a reaction for it to occur. As a matter of fact, you guys remember this at the beginning? Here was the ball. Here was the athlete that was pushing the ball. That right there is a little bit of EA, pushing it and then making it happen. Same kind of concept. Here's what we need to do. Enzymes are proteins. We know what proteins are that lower, I should say specifically, oh no, I'll change it, that lower specific that lower EAs. And I want to let you know that this is specific. So you've got one enzyme or one type of enzyme that lowers specific reactions, the activation energies. And what we do, effectively, this speeds up reactions. It makes them happen more frequently. So it speeds up reactions, and we call this process catalyzes reactions. And in lab, of course, you're working on something called the catalase lab, or the catalase enzyme. So you should be getting familiar with that. Let's take a peek at, get another color and show you what I'm talking about. So this is the energy of activation. It's not enzyme catalyzed. Let's draw a much more shallow hump. So this is enzyme catalyzed. It's lowered the activation energy, has lowered E sub A on the graph. So that's what we're talking about. I should draw the way it could work in an endothermic reaction. Here's an endothermic reaction, and endothermic reactions themselves can have EAs. So that could be the EA, and the enzyme can lower that as well. So there's the enzyme catalyzed for an endothermic. So there we got it. Let's take a look at some enzymes. More things with enzymes. It's a protein. So I want us to go back to our concept of what proteins are. Here's the protein review stuff. Proteins, remember, primary amino acid sequence. Secondary structure was alpha helix, beta pleated sheet due to H bonding. Tertiary, star. Final folded shape. And of course the quaternary had to do with whether or not you had multiple subunits or not. But it's this final shape. I said this for proteins. Shape determines function. You remember this. And let's take a look at enzyme shape. So here's some, some general enzyme shape features, or shape concepts. First off, the general shape, it's kind of a globular protein, some kind of globular protein, and it has pockets, you could call them, or clefts. If you took a look at if you made a model of an enzyme with Play-Doh, and it was kind of this globular ball of Play-Doh, stick your thumb in it in a couple of places, and let's call those pockets. And those pockets are going to be specific kind of lock and key spaces to fit what's going to be aided in the reaction. So they have these pockets or clefts, and these pockets or clefts collectively have a very specific shape, specific charge / partial charge kind of environments or landscape, charge or partial charge landscape. So these guys are going to have certain aspects in these clefts. And what we call them, they are called active sites. These active sites are going to bind to the substrate. They bind what we call substrates, and substrates are basically the reactants of the reaction. Okay, so that's our whole concept. When a substrate is bounded to an enzyme's active site, so when these things are all together, we call this the enzyme substrate complex. So an enzyme substrate complex is an enzyme, a ball of Play-Doh with a smaller ball of Play-Doh, the substrate, bounded or bonded to or stuck into the active site. That's the enzyme substrate complex. Generally speaking, let me show you what happens. The active site, basically stresses, puts bend, but torque, stresses covalent bonds of the substrate. Let's take a look at this. So, how about this? Example. Enzyme catalyzed hydrolysis. Oh yeah. Remember that guy? Hydrolysis? Enzyme catalyzed hydrolysis of a polysaccharide. Wow. This is like old time stuff for you. All that stuff that we've talked about. So here's our cartoon of the part of the polysaccharide. And of course the polysaccharide, I should probably draw it simplistically with our little bond here. And so we take a look at this. Let's put some of this in the middle of an enzyme. So the enzyme I'll draw in green here. The enzyme is this big blob with an active site that looks like this. So here's the enzyme. There's the polysaccharide. And all of those great things that we talked about in this class. Partial charges. If this was a full glucose, you guys would be drawing all sorts of partial charges. You remember all the hydroxyl groups coming off of glucose? So we have partial charges here, partial charges here. So we're going to have like this love fest, this suction love fest based on the attraction of opposite partial charges. And it's going to pull this molecule down, but look at the shape of this cleft. This active site is a big fat honking ridge in the middle. And for our concept, when this thing forms an enzyme substrate complex, it's going to look like this. Excuse me, I'm not drawing the enzyme like I did before. So there's our enzyme before. Now this whole thing is sucked down into the pocket, and it looks like this. Needs to be here. I got to draw that thing a little higher. So big stress get my red pen out. Stress. We bent that bond because the whole thing is being sucked down into this active site. And that, of course, is what's going to cause this whole thing to break. The rest of them are still out here. This goes on in your mouths all the time. Eat some taters. Eat a starchy food. This could be starch. And you've got an enzyme called amylase. This could be, for this example, amylase. Conceptually, amylase is going to suck down and put stress on this bond, and of course the result is to break the bond, and you end up, whoops, let's see, running out of space for my enzyme, but that's okay. Boom, out from here. Maybe we should stick blue there to show the hydrolysis. And then that guy takes off, this guy takes off, and it's ready to break down another. HO. So there's our hydrolysis part right here. That whole molecule is shown with the H and the HO, and the rest of the story you can draw just to keep your picture kind of representative of what it was. Look at that. Look at that. That's amazing. So now the enzyme is, the work is over, the reaction is complete, substrate. These are now products, by the way. So it's, let's identify that. This thing here, starch, is a reactant, and it's a polysaccharide. Then these two things here are technically the products of the reaction. This reaction isn't just going to happen that easily. It has to be aided by the stress of the enzyme substrate complex, the stress as the substrate is pulled into the active site. And that's that. Let's label this. So this is technically, therefore, by our definition, enzyme substrate complex. There we have it. Enzymes make things happen. As I mentioned, go eat a bag of tater chips, and those starches are being broken down into glucoses right in your oral cavity. It's called salivary amylase. It comes from your saliva. So we're actually starting to digest these carbohydrates before they even get down your tube. Good stuff. Excellent stuff. So, we need to take a look at a couple of other neat things. Let's take a look at where enzymes can be found. So, one thing could be actually secretory. So secretory enzymes, just like I talked about, amylase. By the way, good grief, I have left out an extraordinarily important thing. The ASC ending is, in proteins, this is kind of the standard ending for enzymes, enzyme names. Standard ending for enzyme names is ASE. Always remember that. Okay, secretory enzymes like amylase. So here you have something that went through all the great stuff that we talked about in cell biology. We had remember the business. We've made the primary structure, the amino acid chain. It's been folded Think of things like endoplasmic reticulum and Golgi apparatus. As a matter of fact, think of transport vesicle to Golgi. Think of further shaping, folding of protein in Golgi, and then the pinching off of a secretory vesicle that travels through the cytoplasm. Lava lamp, remember, fuses with the membrane, dumps contents to the outside. That is secretion. That's an enzyme released from the cell, and amylase is an example of that, as I just mentioned. So that's one particular category. The next category would be basically in the cell. You could have them in the cytoplasm of the cell. So free in the cytoplasm, I guess I should say. We're going to discuss a famous enzyme that does the first thing. The first demolition crew for glucose is free in the cytoplasm. It actually takes glucose from being a 6-carbon molecule to 2 3-carbon molecules. Boom, in the cytoplasm. Then we're going to actually have, I guess we'd call this cell. We could have those that are in membranes. So we use the word integral in membranes. So this is an idea here. You guys have drawn this before. We just did a whole bunch of this stuff when we did chapter 7, but I want to remind you that, we could have an enzyme right here with an active site. So here is a phospholipid bilayer, as we've seen before. Boom, there is our integral membrane. This could be an active site. Good stuff. So that's integral to membranes. Moving down, other things that we should discuss is a lot of times for integral membranes or the integral enzymes in membranes, a lot of times you could have simple ones. Simple basically means one enzyme and one function, but we could also have what we call multi-enzyme complexes. That could be something that we see in multi-enzyme complexes. Let's take a look at this. Here you actually have your book will bring you to an example of one called pyruvate. That name is going to become familiar to you in the metabolism chapter. And then dehydrogenase. Pyruvate dehydrogenase is an enzyme complex. It actually catalyzes 3 sequential reactions. So pyruvate dehydrogenase catalyzes 3 sequential reactions, meaning that the product of the first is the substrate for the next on down the line. And that it has multiple, this complex is basically made of multiple copies of 3 different enzymes that are all clustered together. Basically, it's massive. It's a big pyruvate dehydrogenase complex. That's what it is. Okay, let's see. I think that's, I'm going to leave that, and now I'm going to move to the concept of enzyme inhibition. Okay, so here we go. Enzyme inhibition. So you've got this great, big, awesome protein, and it doesn't necessarily need to be active at all times. There's so many ways that enzymes can be controlled. I mean, that's a whole side discussion. It's pretty intense. So I'm going to stick with the basics here. And we're going to deal with enzyme inhibition and activation. And I'm going to use a very simple example that's in your book. I'm going to draw an enzyme and show how things can happen. So draw it like this, of a little pocket down here, and something like this, and a step here. Okay, so this is going to be our enzyme. Oops. There's our enzyme, and what we want to identify on our enzyme are a couple of features. Here would be our beautiful substrate. And I'm going to identify a couple of places here. This is, I shouldn't use green, that's kind of a wrong color for this. This is what we're going to call the allosteric site. So I'm going to stick in something. Scenario 1 is that right here I've got something that has bonded to this thing. This thing is called the inhibitor. As a matter of fact, I'm going to give you the specific name. It's called the competitive inhibitor. So the competitive inhibitor binds to the active site. Very important to do that. Binds to the active site, and it prevents, therefore, the substrate from binding. That's scenario 1. So as drawn, this whole thing here, let's see, let's go from here. This whole thing I'm going to title competitive inhibition. Okay. Now I'm going to use the luxury of my board here for the last thing to say. Let's take out this next example, this part, and what I'm going to do is change the enzyme, is I'm going to stick in something here. This is called an allosteric inhibitor. And when that thing binds, it changes the shape of the enzyme. Here's the way the enzyme used to look, right? But now, because of the allosteric inhibitor, it's kind of sent a shockwave through the entire enzyme and changed its shape. And now the substrate can't fit. So, allosteric inhibitor binds to allosteric site. Shape changes. The shape of the active site changes, and therefore substrate can't fit. Okay, this one is the second and the final thing for this lecture. Is going to be called allosteric inhibition. So, a couple of different ways then. Here's these enzymes living out there in the world, wherever they are, whether they're in membranes, whether they are free, and you could actually have a competitive inhibitor, type 1, that I've already erased, that keeps the substrate from binding. That's going to prevent that reaction from happening, or at least slow it if you keep those enzymes bound up. The other one is allosteric site, where another product is actually going to change the whole shape of the enzyme and make it non-functioning, because it can no longer, as long as it's bound to this allosteric site, it can no longer bind the substrate because the pocket has changed. So those are the ways that enzymes can be controlled, at least on a very graphic and big-time level. There's a whole bunch of other ways as well, and it's just scratching the deep and awesome surface of cellular biology. So this is where we'll close this lecture. There's one more thing for me to talk about with enzymes before we go straight into the wonderful world of Chapter 9. Please look at this lecture and take notes as if it were a class. Detailed, good notes. Look up things in the book if you need clarification, because we're going to march forward, and this is getting progressively more and more intense, but really, really cool. All right.